Brian Smith from Arizona State University. And that kind of I think it's very much related to the previous talk about energy efficiency and kind of optimal computation, because Brian has been studying insect brain for a long time, and insects obviously very efficient systems using very little energy to solve very complex tasks. So I had the pleasure to collaborate with Brian for a long time. And so Brian, please, this microphone is yours.
Yes, well, thank you, Maxim. As Maxim said, well, it's my pleasure first to come and talk to you about something that both Maxim and I have been very much engaged with for quite some time. In fact, I think we've been collaborating on various projects related to this, and I'll touch upon some of them for 10 years now. This is a bigger project, and toward the end, I'll talk about the program that was funded by DARPA on miniaturization. But the first thing I want to do is try to set up for you the biological problem that we've been working on. I want to then talk about how plasticity in a tiny brain, like an insect brain, a fruit fly, or a bee, in some sense has evolved, even in the periphery and early sensory processing, to solve this problem. And then I'll talk about how the brain has adapted to these problems when the brain evolves to something that's a fraction of the size of even a honeybee brain.
Brian, so could you put it in presentation mode?
Oh, I'm sorry, it's not. Yes, I should.
Thanks.
There, that's probably better.
It actually didn't change. It looks like maybe a wrong button.
What didn't? Oh, I know what happened. Hold on, I'll have to share my screen again, I think. Sorry. Now it should be in presentation mode.
Yeah, thank you.
Okay, so as I was saying, the general problem that we're working with is a problem of olfaction and odor identification. The models are various kinds of bees, here a honeybee and a bumblebee looking to visit flowers and identify flowers for the nectar and pollen or carbohydrate and protein resources they need to collect to survive, or a fruit fly looking to feed or lay eggs on a piece of fruit. Now, they might use many different kinds of cues from different modalities, but in terms of olfaction, honeybees and fruit flies use odors to identify flowers that are in the correct state for them to visit, or in the case of fruit flies, looking for fruit in the right state of ripeness or not ripeness, rottenness to lay eggs in. Now, these odors are almost invariably, as you may well be aware, not just one or two odor components, but they're made of many different types of components shown in these complex mixtures of this is a flower called brassica, and I'll talk quite a bit about these flowers in a minute with bees. Or with strawberries, you may get up to anywhere from a few dozen to a couple of hundred different chemical components that all are important in helping the animal identify the flower. So in the sensory systems we've shown, and I won't go into this, both flies and bees can identify almost all the components in this complicated brassica or this complicated strawberry mixture. So now the problem in identifying the flower is that the flowers can vary from one to the next in terms of the precise composition of those components in the mixture. And here on the right, this is a principal components reduction of probably a 15 or 20 different component chemical mixture in snapdragon flowers. And each of these small yellow dots or orange dots or the small green dots, that represents an individual flower and the particular mixture of odors that it gives off. And the cluster of points here represent flowers in one state, and what that state can be, I'll get to in a second, versus the green ones here are flowers in a different state. And note that the precise chemical composition, what is in those mixtures, is the same from one of these clouds of points to the next. The only thing that differs slightly is the ratio. So now let's say you're a bee and you have to identify nectar from these orange flowers and reject these other flowers over here that don't have nectar. So you don't want to waste your time with the green flowers. You have to identify the ones that are orange. Well, how would you do that? Well, the problem is not necessarily the mean odor composition. Yes, on average, the orange are different from the green, but the bee also has to have some kind of concept of the variance. And one of the things I'll show is that bees are very sensitive to the variance, and and learning about the variation can help bees solve this classification problem. In other words, is this new flower similar, not the same as any of the orange flowers that it found, but is it similar enough for me to classify it as the same thing? And many of the kinds of plasticity, and I'll talk about a toolbox of plasticity in the bee's early sensory processing in the brain that it uses to help solve this classification problem. And my colleague, Tanya Sharpie at Salk pointed this out to me a few years ago that is very analogous to a problem in vision. We can all look at this statue and recognize it, even when that statue appears in slightly different orientations or slightly different visual situations. However, if I give us a slightly different statue, we immediately recognize it as something different. So the analogy between this visual problem, what I'm talking about is if you think of, here's that cloud of points that I just showed you, and remember each of these points is a reduction of a chromatogram with about 50 different components. If we imagine now that all of these faces belonging to the same statue belong to this cloud of points and this different one belongs to this one, and the bee, just like our visual system, is trying to separate these clouds based on meaning. Now, a natural problem for the bee is, in terms of meaning, is that these odor compositions reflect pollinated and unpollinated flowers. The bee wants to visit unpollinated flowers because those are the ones giving it nectar and pollen that it needs to collect. And once the flowers have been pollinated, they'll change the ratios of these components to be different. So these clouds in a natural problem for bees could be unpollinated versus pollinated. For a fruit fly, it could be fruit in the correct ripeness state, fruit in a very different kind of ripeness state. To show you that these mixtures many times do actually mean something important, if we can analyze now, these are some of the components here, these chemicals in a brassica odor mixture. This is a list of them, and this is a principal components analysis of the variability from flower to flower in these components. And you'll see on this first component, it's several of these odors load significantly. So variation on this component involves a correlation between these 6 different chemical components of the brassica mixture. If we now correlate the score on that principal component with things out at this end, they have a lot of these 6 components. Things at this end, they have little of these 6 components. And we look at how much pollen does the flower have. That correlates very nicely with a pollen content. So a lot of these components present in the mixture says pollen to the bee. And if the bee's looking for protein, it's important to pick out these 6 components. A different set of 4 components now involve nectar. and this was principal component 2, or in this case, I'm showing principal component 3. The score on this component, which would mean a lot of this component called PAA and very little of these 3, or vice versa. So the contrast of these components mean a lot of nectar at one end versus very little at another. So this complicated mixture can be reduced to something a little bit simpler in terms of signaling to the bee. So if I plot these 2 principal components against one another, what this chemical mixture is able to do is separate 4 different kinds of flowers for a bee. Flowers that have pollen and nectar, flowers that have only pollen, flowers that have only nectar, flowers that have neither. Now this space is a little bit too sparse right now, and we're trying to supplement this with some new work we're doing with brassica and other flowers. But generally, this is the idea, that this mixture can signal several different things using these submixtures that vary from flower to flower. Okay, so a lot of what I've done over the years and working with Maxim on these projects is to look at how early sensory processing can help manage this odor complexity. A honeybee antennal lobe, a honeybee itself, well, you may have heard mice have about 1,000 odorant receptors. Humans have about 300 or 400. Honeybees have about 160, and the combinatorial ability of a space with those dimensions is enormous in terms of the kinds of odors that we or honeybees or other insects can encode. And what I'm going to argue is that for a honeybee, this enormous coding space is not used most of the time, or most of it is not used, simply because what's important to the bee at any given point in time is a very narrow range of odors. And a lot of the plasticity that I'm talking about is how this space can be, in some sense, reconfigured or warped to help the bee solve a particular problem. So this is a honeybee brain, and coming in from the antennae here, where there are along each antenna about 50,000 or 60,000 odorant receptors, expressing, as I said, about 160 different odorant receptor neurons, expressing about 160 different receptors across those 50 or 60,000 neurons. They project their axons into the antennal lobe at the base of the brain right here, where olfactory receptor neurons expressing the same receptor project to the same glomerulus. So there are about 160 glomeruli. This is the same story that you may have heard for the mammalian olfactory bulb. And what I want to emphasize here is that if you take any of these glomeruli in this cross section, and you magnify it, here in yellow, you see the sensory axon terminals, the olfactory receptor axon terminals. In blue are dendrites of projection neurons, mitral cells in the case of mammals. And interspersed throughout this glomerulus, you see a number of different modulatory neurons or representations of them. An octopaminergic neuron that has spreads its axons here in the cortex of this glomerulus, potentially able to influence the interaction of the axons of olfactory receptor neurons with the dendrites of projection neurons. But you also see serotonin, you see dopamine, all having patterns of expression in the antennal lobe. Now I want to focus a little bit on the octopamine here right now. This neuron is called VUM MX1. It was discovered probably 30 or 40 years ago now. It's a cluster of neurons that sit here at the base of the brain and receive input from sucrose-sensitive taste receptors. And when the bee gets sugar, it releases octopamine all over the brain, here in the mushroom bodies, up in the upper part of the brain, as well as, and I'll say these mushroom bodies are multimodal convergence centers for different sensory modalities. And I'll talk more about them later. But this is the neuron that you see releasing octopamine whenever the bee's feeding on sucrose, and practically every glomerulus here in the antennal lobe. So one of the nice things about bees is we can bring them into the laboratory and train them using Pavlovian conditioning. We just give them an odor and follow that odor with a little bit of sucrose reinforcement. And after one to a few trials, the bee will stick out its mouth parts here to the odor alone. And this is Pavlovian conditioning. And it's a way that we can assay how the bees are learning about odors, how well can they detect odors, how well can they discriminate odors in a very controllable laboratory paradigm. And this just shows you a well-trained bee responding to an odor that it has learned to associate with sucrose, with extension of the proboscis. So all the behavioral responses that I talk about here in a minute will just simply be proboscis extension. There we go. So this is what a training protocol looks like. On the first trial, very few bees out of 20 or 30 or 40 respond. After 1 or 2 or 3 trials where they get sucrose reinforcement with the odor, you'll see they start to respond. So after about 5 or 6 trials, we can usually train most of the bees in any given group that we're selecting. Now, when we take the bees and test them with the odor they were trained to, many of them respond. And if we start changing the chemical nature of the odorant by changing the carbon chain length, by changing the functional oxygen group, you can see this generalization gradient that reflects the perceptual similarity of this odor to the bee, to the odor that it was trained to. And this corresponds very well with patterns of activity that we see in the antennal lobe, and I'll talk about those in a few minutes. And we can change the training paradigm to make it more difficult or less difficult here. Training to a pure odor followed by sucrose reinforcement, and then we train it to a different mixture of a different odor. So this is 10 parts odor A, 0 parts B, 0 parts A, 10 parts B, followed by nothing. And if we decrease the concentration so that we can make the conditioning protocol very different. And afterwards, we see this nice generalization. This is the odor it was trained to. This is the odor it was trained, it does not have sucrose. And when we change the ratio of those odors, 9:1 to 1:9, again, we can establish these generalization gradients. So these are some conditioning protocols I'll talk more about in a minute. And the last one I want to talk about is learning to ignore stimuli. It's called latent inhibition. If we give the bee presentations of an odor followed by nothing, and we do that 40 or 50 times, and then turn around and try to train the bee to this odor, now followed by sucrose reinforcement, they'll learn this odor slowly relative to a different odor where they have no experience with that odor. And here's the blue odor, here's the orange odor. They learn it slowly because they've learned to ignore it, and they have to overcome this learning in order to effectively respond to it. I put this in because there are many different components. There are visual contexts that I'm not going to get into today. So how's this learning encoded in the antennal lobe? This is the bee brain. This is the antennal lobe. And what we do is we load the axons coming from the projection neurons out here into these areas called the mushroom bodies. We can put a little bolus of fura, a calcium indicator, in these axons, and it'll backfill the axons into the antennal lobe, and we can visualize with our imaging system maybe the dorsal 20 glomeruli here in the antennal lobe. What that looks like under the scope is there, and we can identify these glomeruli from individual to individual, and you present an odor to the bee, and that's what the activity pattern looks like. There's a spatial property, certainly, and there's also a temporal property here. So if we go from that one pure odor in that behavioral paradigm I showed you, this would be the 10-0 plus, for example, to the other pure odor, the 0-10 minus, and we start changing the mixture 9-1, 7-3, 5-5, 3-7, and so on. You'll see that these mixtures grade into each other. And if we look at 100 millisecond time slices, you can see that the odor, for example, this would be the pure hex, will start to evolve out along these transients, and the odor goes off and it comes back to baseline. This is the 10-0 odor evolving out, coming back to baseline. This is 10-0, 9-1, 3-7, 5-5, 7-3, 1-9, 0-10, and you see these transients just march across this space, and they represent in this coding space a perceptual dimension that encodes these different odors. So now what I want to do is talk a little bit about these mixtures, and we didn't talk about these big orange dots here earlier, or these big green ones. And these are artificial mixtures that we make up in our lab to span the natural variation in this space encoded by these natural odor mixtures. We're going to start testing the bee and training the bees to these natural odors, now training it to the variance. And just to show you, these are 6 different components. The bees can detect all 6. You can see that the mixtures will be similar to each other. And here's the orange cloud of points, I think. Here's the green cloud of points, those 6 mixtures that span these dimensions. And now all we're going to do is train the bees to discriminate the orange from the green. And under normal situations, if we look at the antennal lobe and we correlate the response in the antennal lobe to all of these 6 orange mixtures, you'll see this cluster of correlations here that represent reasonably closely correlated responses. We do the same to the green cloud, and then we do the same across the 2 clouds of points, and the correlations within the green are strong, within the orange are strong, and naturally they're weaker going from orange to green. Now, if we train the bees to discriminate orange plus, green minus, what happens is the correlations within the clouds become a little bit lower, but the correlation between the clouds really become lower still. So what's happening is that space in this antennal lobe is becoming, this is where I say it becomes warped in a sense, to better represent the differences between these odors that are suddenly important to the bee. This is now associative conditioning and how it's represented. And this is something I've worked on with Maxim now, is that non-associative learning that I talked about. And these are these transients to 2 different odors. And the green is the 50-50 mixture. And when we say generate latent inhibition to the blue odor here, and the red one is the novel odor, what happens is I don't get so much of a shift to these odors, but rather the mixture now shifts away from the odor the bee's ignoring and becomes biased toward the novel odor. So this is another kind of non-associative plasticity that's present in the toolkit in this antennal lobe to analyze this natural variation. And we've done some modeling. This is some modeling we did several years ago. We tried to predict which synapses, just using Hebbian learning to modify the synapses. We could identify what types of synapses most likely would give rise to this effect. And interestingly, after a computational model identified the synapses that give rise to our biology, some people in fruit flies went and modified those synapses and showed that yes, indeed, they do produce this effect. I would like to have done that myself, but it was not. But in fruit flies, you can do some things that we still can't do with bees. And lastly, I'll skim over this one. I won't talk about this so much right now. This is a novelty effect. With that novel odor, it turns out that the bees use their antennae to tell us what they think is novel after they've had exposure to an odor for 50 times. When you give them a novel odor, they really pay attention to this novel odor. And this novelty effect, I'm going to skim over this so I can finish on time here. This novelty effect we've shown in some work now with Maxim is encoded in the antennal lobe. When you present them a novel odor, both projection neurons and local interneurons increase their responses to that odor relative to control at the beginning. So to conclude this part and segue into the miniaturization issue toward the end here, the case that I'm making is that there are several forms of plasticity that exist early in sensory processing that's helping the bee solve this classification problem with floral odors to figure out which are the right kinds of flowers to visit. One of the things that we've worked on now with DARPA funding with Maxim, as well as with some other colleagues, engineering colleagues, Ted Pavlik, Kevin Cow at ASU, and I won't talk about the modeling or the engineering with this either, is to look at what happens when the brain becomes smaller, when it becomes miniaturized. And to do that, we're looking at stingless bees in the tropics. And there are probably about 1,000 or more species of these bees occurring in the New and the Old World tropics. And they range in size from a honeybee, about 100 milligrams, to something the size of a fruit fly, literally down on the order of 2 or 3 or 4 milligrams. This is a picture of one of these bees that's not even the tiniest, and this bee is about twice the size of a fruit fly. We can look at their brains and look and see now what happens to their brains in terms of investment in processing as we evolve tiny and tinier brains. And I'll say that this evolution of miniaturization happens multiple times independently. So it's not just we see it happen once and we see some way that the brain changes, but rather when it happens again and again, is this change now consistent? And I just put this movie in to show you what these bees look like. All those hovering bees are guard bees. And the size of that nest entrance is a little bit less than the diameter of a dime or a 1-cent euro coin. And this is the head of a pin over here, if you want to get the body size approximately right. They're very interesting bees to work with. And if you think that they are good because they're stingless, I have another video right here, and that's what they look like when they get mad. So if you're around these bees, you'll be picking them out of your hair, out of your eyes, blowing them out of your nose for a few days afterwards. They can be quite interesting to work with. So what we've done is we've just simply estimated the volumes of all these different areas. And here, these are those calyces, the multimodal processing centers, high up in the brain. These are the antennal lobes, and here are the optic lobes, the visual neuropils, the compound eyes of these bees. And we can estimate the volume of each of these regions in the brain as the body size of these bees begin to decrease. So this is the log brain region volume. This is the log body mass. And in big bees, you see, reasonably good size optic lobe. These are the visual neuropils, and the red shows how the visual neuropils change the slope as the body size decreases. And the important point here is that for the antennal lobe, the volume of the antennal lobe in green, and these are different areas of those multimodal processing centers, the mushroom bodies, they also decrease in size, but at a much lower slope. So what that means is that bees, as the brain gets tiny and you put more emphasis on certain kinds of processing, is you're preserving the relationship between the antennal lobe and the mushroom bodies, and the antennal lobe and all that plasticity I just talked about, and you're sacrificing to some extent your dependence on the optic lobes. So at least we're able to see how some of this plasticity is affected as bees evolve with small body size. This is just a summary that in terms of the investment, relative investment in the antennal lobe, and the decrease in investment to the optic lobe as the bees change in size. So I think I'm about at my 30-minute limit. I just want to emphasize a lot of people we've worked with over the years, people who've been involved in both the biology, Fernando Locatelli, a former postdoc now in Buenos Aires, as well as Maxim, and Kevin and Ted, who were involved more on the miniaturization project. Okay, so let me end it there. Thank you very much.